So thank you for this chance to be here, talking with you about super resolution and what we can do, or we can try to do, with super resolution. So the title talk will be devoted to super resolution fluorescence microscopy mainly. Just this spot, because maybe you can be interested in some book on this. This is the newest, and in case you're interested, you can write me. I can also send you the PDF in case also of these ones. A second spot, because in Trieste there was the Festival of Science Fiction, and in Genova is 13 years we have Festival of Science. We are also looking for people working with us. And so in case you're interested, you can go through the website and telling us that you can help for this event. Now, I come from the Istituto Italiano di Tecnologia. That's in this not very nice building here. But what I have to say is that inside you can find, as probably here, something related with multidisciplinarity. So we have a lot of disciplines that are converging all together on some tools. One of them is the robot. I will tell you something later about the robot. But this is my former group. You have known yesterday Davide Mazza. We come from Department of Physics, University of Genova. Then now Ilaria Testa is in Stockholm with an ERC. Paolo Bianchini is in IIT with Giuseppe Vicidomini that is back from Hell Lab. Francesca Cella is back from Stelzer Lab. Valentina Caors is in London Imperial College. Emiliano Ronzitti in Paris. We started in 2003 with them, 1982 in microscopy, and this is the current group, and we have 47% of women in our group. Let's have this number growing, including the robot. The robot is neutral, so we don't know. You can split there. And we have some friends that probably you know. This is a picture taken the day he got the Nobel Prize, the day after a great night in Göttingen. In Genoa, as probably you know, we also have an Icon Imaging Center. There is a center where we work at different levels. So we have, you can come to the Nikon Imaging Center to check newest Nikon instruments for focused application. We have a research and development group as well, and some original setup that you can use, and also some educational activities. One of them will be in December, the second week of December in Genoa. And again, ICAB. Maybe you have seen this guy, these talents, Italy Got Talent. I don't know the name of this. But this is more or less the attractor of all our activities, including microscopy. Our final aim with the microscope, development of some microscope, is to bring to the tip of ICAB a portable microscope, allowing molecular identification and detection. So it's not only for fun, it is in the lab. And in this lab, what we do is mainly developing instruments, new instruments. Nanoscale. Super resolution deals with nanoscale, because with a microscope you are able to get information to 100 nanometers, more or less. And now you can move down to tens of nanometers. In principle, resolution you have, we will discuss about this later, and we can discuss also in the lunchtime if you want, is theoretically unlimited today. Looks strange, sounds strange to listen to this, but today, spatial resolution for the microscope, optical microscope, is unlimited. And so when before, from chromatin DNA, you had something like this for nuclear pores, today you can get this kind of information. The idea of improving the microscope comes from a seminal work by Richard Feynman. I don't know if you know this guy, Nobel Prize in Physics for theoretical physics, but in 1959, he had a great talk at Caltech. And during this talk, he defined what nanotechnology is. So it's our ability to control and to observe at the nanoscale matter, nothing more. That time, one of the systems he was interested in was the biological system. The idea that in order to understand, you simply needed to have a look and then getting your final decision, at the nanoscale started there. Unfortunately, the scale you're interested in is not compatible with the scale of the conventional optical microscope. You are interested in protein trafficking, small object, and you have an object that is able to visualize a lot of them. You could go to the electron microscope in case. And so you could say, so why are you pushing the optical microscope? Because if I want to get the information at the molecular level with the electron microscope, I have to ask you to be very kind. I have to ask you to be very kind and to solidify you, to physically cut you, to embed with metals, and then to shine some electron beams on your slices. I don't know if you agree in having this approach in order to study what's going on. In case you have it, wonderful. However, I would not be happy because I want to follow what's going on in time. And the optical microscope is unique in this. This is the reason why people is pushing with the resolution and the optical microscope. And so this was the final sentence by Feynman. And you know that before, with the optical, you could get this information. Today, it is one. This is the nanomachinery we are interested in. Is a real nanobiomachinery because small changes at the nanoscale influence what's going on in the micron scale, 100 microns, centimeters, meters. Perfect system. And if you have a look here, I like to bring your attention chromatin DNA because what I think about super resolution methods is that they are unique not only because they allow you to get nano information with some light and some curved glass, but also they allow you to tune in resolution. And this is very important because you see that here you could have different questions at different spatial scales. What is missing today is some part of technology, and we will discuss about this, but also sometime what is missing is the biological question, the real biological question. When you have your problem and you face this problem with an optical microscope and you are limited in diffraction, are you sure that you have a question for which having a better resolution can allow you to conclude something? If yes, move to the super resolution field. If not, continue with your wide field microscope using some other biophysical tool, I don't know. And so this machinery is fantastic, you know better than me. It's like being, I imagine that there is a library and there is a worker inside, and when you're passing through and you're running, this guy is getting out, bringing you a book open in the right page. So it's something that is amazing. And so the mechanism at the nanoscale is very important for understanding how this system works. And this is one of our biological interests. This is the reason why I'm showing you this library with all these activities like the nucleus, within the nucleus of the cell, and the possibility of using the microscope at the nanoscale to decipher something there in this mechanism. And so now is relevant moving from this image. Just have a look to this image for understanding something more, maybe. Now I have another question for you. That's only for you, for biologists. Is in order to understand something about what's going on with the nuclear pore, I have to use labels. But these labels are, in case they are green fluorescent proteins, they are 27 kilodalton that I place there. Do you think about this when you design your experiment, about and related with your question? So this today, that you have this very good resolution, is a crucial question. Before you could escape, not today. This is my mind. So with a light microscope, you know you are here. With your eye, you are here. With the optical microscope, you are here. and then before you missed that. And even if you have this drawback with resolution, if you go through Nature Medicine, you see that 80% of papers deals with light microscopy and 20% with electron microscopy. And if you have a look again to optical microscopy, you see that we are moving to this direction here, increasing resolution. And we use visible light. Particularly interested in using visible light, that is from red to blue. I will be back on this later. And fluorescence, that you know very well. Well, you know very well. Not sure that you know very well fluorescence. Or maybe you do. But the main point is that today, most of the times, what we do with fluorescence is collecting the intensity information, nothing more. Then we complain when we have bleaching, but mainly we deal with intensity information. That is not only the unique information we can use. We will see how to use other information. However, specificity given by your labels is great for studying something happening within the cell. How to excite fluorescence? I don't know if you know about two-photon excitation, if you had something about this, but you know that you have two ways. Normal way, so you need energy 10. You provide energy 10, your molecule is in the excited state, and since it's a fluorescent molecule, it relaxes back emitting photons at a wavelength that is longer than the excitation. It's possible to play the very same game, bringing the molecule to the excited state, 10 of energy required, using photons having an energy that is 5 plus 5. That is 10 at the end. And so you can play this game, is an ability game like this one, and you can bring the molecule to the excited state, and when the molecule is in the excited state, it's done. Then fluorescence is the same. Unfortunately, the temporal window to perform this action is 10 minus 17 seconds. This is the reason why it's not so common to have this effect. The only way you have to improve your ability in getting this event is increase the number of photons at the molecule, which is, in your microscope, the region where you have the highest density of photons. If you think that you have light, your circular lens, then light is making something like this. Density is number of something divided by area. Area at the apex in the focus is minimal, and so here is the maximum density. In fact, when you have this process, this is the conventional excitation, you excite everything here. With a 2 photon, you excite only here. That's great. You are excluding a lot of information, spatial information, in this case. With your optical microscope, you can perform 4D. That means 3-dimensional and time that you cannot do with the electron microscope. You add this object, that is great, an intrinsic marker, fine. This comes from Martin Chalfie. But what was really relevant for super resolution methods was a seminal paper by these guys, 2002, demonstrating something that people already knew about green fluorescent protein, about switching of the emission wavelength or changing of the emission properties. But in this case, they brought to our attention the fact that there are photoactivatable proteins, green fluorescent proteins. What does it mean? They are expressed. You shine a light for exciting them. You see dark. Too bad for people coming from Genova. All this energy lost. Doesn't matter. But then you shine another light, some other energy. You photoactivate, and then you can see them. This is great, because you can select when you have the expression, where, and this is the protein side, but then you can decide by your own, with your optics, when you want to see them, in case they are in a special place, in a particular place in the sample. This is what they did. And if you move to 2-photon excitation, since the event happens only in the focal region and not everywhere along the excitation pathway. You can very precisely photoactivate molecules, for example, here, and then you can see some trafficking from this region to the membrane, and you are sure that the fluorescent molecule that then you find here can come only from this point. All the other regions are not involved. And if you want to photoactivate in your cell, you can photoactivate in a specific region in the 3D space. You have no doubt the region that is originating the signal when you are using 2 photons. And again, you can work in time. Visible light. Visible light is the reason why we are diffraction limited, and the reason why I'm studying with the optical microscope. This is my team, red and blue, visible light from red and blue. This is my scientific reason for working in the visible region of the electromagnetic spectrum. But these 2 wavelengths, so red and blue, are from red around 600 nanometers, blue 300, 400, whatever you want. Is not relevant being precise, but then your resolution is this one. You cannot escape. And if you go to Jena, they don't have this monument, they don't take too much care of this monument. It's not very cleaned, let me say. You will see why I'm saying this later. But you can find written on the stone this law. And this means that you can do whatever you want, you can tell whatever you want, but you cannot overcome this. Despite this, today, D is moving to 0. So it's unlimited. What does it mean being limited? With sound, we know this very well. If we think about sound and about frequencies, we can select high frequencies, low frequencies. Our system is a system cutting frequencies. And the microscope is the same. You insert a lot of frequencies, and the microscopes cut the frequencies. Frequencies are variation of a signal. In time or space, doesn't matter. But the microscope cuts them here, more or less 1/2 of the wavelength. Or if you want to be sharper, using this relationship. You cannot do anything after this except super resolution. And super resolution means that if you place your fluorescent molecule at distances closer than the diffraction limit, this is the best you can get, you could get before, and this is the best you can get today. I like to bring to your attention this guy. I don't know how many of you know this guy, Giuliano Toraldo di Francia, 1947. He introduced the term super resolution. Which was the meaning? The meaning was, if you are able, you collect this information, you have this frequency cut, if you have some information about your system that allows you to have an analytical continuation of this curve beyond the cutoff frequency, so you can put something here, and then you are back to space, from frequency to space, you are in the super resolution domain. Dot. And this was one of the seminal works by Giuliano Toraldo, this one 1949 and this one 1955. Later you can find in this paper, in case you are interested in fundamentals by Colin Shepard, distinction about different kind of terms, of meanings for super resolution. In the super resolution, let me say history, you know about the Nobel Prize, we will be back on them immediately, but there was this guy, Mats Gustafsson, that unfortunately passed away too early, that was the very first one implementing with the structured illumination microscopy the Toraldo di Francia idea of extending the domain of spatial frequencies, on your side, and then allowing you to be back to space with a super resolved image. And so what he got, for example, was something like this. So this is the wide field, and this is what you can get applying the structural illumination. It's amazing the difference and the meaning of this kind of images and what you can get from them. But now, let me be back here, your eye, your microscope, and today the Nobel Prizes that are here. Everywhere you want on this side, unlimited. Why they got the Nobel Prize? Why they got it in chemistry? This is the motivation for the development of super resolved, Torado di Francia, fluorescence chemistry, microscopy, an instrument. Yes, they were cutting your hair more than in 4 parts. And I liked a sentence from a paper by Stefanel here, and here you can see what's going on when you apply, when you move to wide field to super resolution. I like, this sentence a little bit below. Diffraction barrier is crumbling. In Italian translation is sgretolata. The meaning is the very first time people is using this for super resolution. Not overcoming the diffraction barrier, surpassing, killing, whatever you want. Crumbling. The meaning is, diffraction limit is there, fine. We do something different to get unlimited spatial resolution. And the game is this one. It's difficult to find the verb now. Overcoming, surpassing are not allowed, so circumventing. In which way? Well, if you are able to preclude simultaneous emission of adjacent, means emitting object at distances closer than the diffraction limit, spectrally identical fluorophores. Spectrally identical fluorophores. If you're able to do this, and you are in the nanotechnology, right? Because now you need to control at the scale very small, nano, something, switching on or off of a molecule. If you're able to do this, you can get unlimited super resolution. And doesn't matter if it's fluorescence. Fluorescence is great for people working in microscopy because you have a very high signal-to-noise ratio. You have a dark background and a bright signal. In transmission, you have a bright background and a dark signal. And then if you want to deal with, and this is absorbing and non-absorbing, if you want to deal with scattering, non-scattering, it's even more difficult. Spin up, spin down is another possibility you have. In order to increase the resolution or your ability in getting details in this room, what can you get without spending money? You look at in this room, and you want to increase your ability in designing details related to one person you are interested in. What could you do? An idea? I'm from Genova, so it has to be at no cost. So please don't tell me switch on light. There is a cost here. I have to move, yes. I'm also lazy. But I can do something like this. Immediately, here, I see better. Then you can argue that you want to see everything. Well, let's scan. Now I have to design my experiment. When I design my experiment, I have to take care of the dynamics of the system. So if you move fast or slow, I can put you in the image never or 2 or 3 times. But this is part of my design of the experiment for a certain question that I have clear. So you can play this game. So it's very easy at the end. In fact, this is your beam. These are the molecules there. And you have 2 methods. One developed by Betzig and Mörner. Let me simplify in this way, if it is not exactly true. and the single molecule, and the other one by Stefanel. In this one, they are so friend with the fluorescent molecules that they can ask them to send the light not all together, but in cluster made by a sparse distribution of emitters. What does it mean, sparse? Sparse means that you can convince your molecule that are dense in your system not to emit all together, but to emit only those that are distances larger than the diffraction limit. How can you do this? We will see. So they are able to convince them to do this, and so they can collect single molecule information. L is German, is more solid on this, and what decide to do? A game like this, but instead of using the end, is using a second beam. A second beam that is convincing the molecule to go to the ground state, not emitting in a natural way fluorescence, but for another pathway that we don't collect, that we don't see. If this beam is very intense, so if you are very strong, the hole at the center is very small, and so you can get information only from a very small amount of molecules. And in this case, you know the position because you are scanning. In this case, situation is stochastic. You will see why. So let's go to the Mörner and Betzig case. Keyword here, the knowledge from Toraldo di Francia point of view is I have my sky, I have a few stars at distances larger than the diffraction limit. I am sure that they are, I'm sure, because I know very well their photophysics, I manage very well the photophysics and everything, so I'm sure that the emitters are single molecules. If this is not true, okay, you are in another field, not in super resolved fluorescence microscopy. But if you're sure that the emitter is a single molecule, what can you do in order to localize the position of the emitter better and better and better? The case is here, let's assume that you say something altogether. Then if all of you stop and only one is speaking and I'm turned, and this guy continues speaking, I can try to imagine where he is, or to localize, because I continue receiving information from one element in time, only from this guy. And this is what happens. So what you can do is that at the very beginning, you know that your molecule is here, more or less 200 nanometers, but then this molecule continue emitting, telling you, I'm here, I'm here, I'm here, I'm here. I know you are there. And then you can scale with the square root of n, where n is the number of photons emitted. Think that when n is 10,000 photons, that is a good number, you are scaling to order of magnitude with the conventional ability in localizing the single molecule. So you go from 200 nanometers to 2 nanometers in localization precision. And so this is the game played in the storm and by Betzig and Mörner. L is playing this game. I will be back on this relationship. But what he obtains, this is only a graphical explanation. It's not true that if you increase the intensity, there is a real shrinking. But the effect is like a real shrinking. If you increase the intensity of this second beam, you can be more precise in getting information from your sample, and so you can improve your resolution. I will be back on this, maybe. And so, if you have your molecules in this shape, they are here. What you get with a conventional microscope is this information, more or less. What you get with these two methods is this information that is different, a different content. Another aspect is that you can count the molecules. So there is a quantitative aspect that sometimes you're interested in fluorescence. How many times you were trying to claim I'm performing quantitative microscopy, and you are using the intensity information to say how many molecules are there, but then there is always something that makes you not so sure that there are 110 molecules there instead of 98. Now you can count them. Again, Feynman. Feynman and this book. I don't like very much what is in this book, but tell something related with Thoraldo again. There is a discipline called psychological optics that I don't like, the term I don't like. But Berkeley, the statement by Berkeley was, when light comes to your eye and is inside, starts a process that is that phantoms that are in you are getting out. As saying that what you see is what you already know. This is what there. And this sometimes happens with the super resolved images. You see filaments and you recognize them. I'm not sure that we are able today to recognize real new objects, but we can train on this. However, it's not only a matter of details. If you like this picture, and this is a detailed part, and this is detailed, probably you also like this. I think, not sure, but I think. The matter is how the data come to you and let you get a decision or another decision because of the feeling they're bringing to you. So if you see this image, probably you recognize Lincoln. But if you have a different view, it's definitely different. Gall at the window. And so your decision can be different. You would look at this for hours and not Lincoln, for example. If you recognize here Marilyn, and if you have a different data presented in a different way, again, the information, your decision is different. Now you can perform like a microscope. If you close a little bit your eyes and you reduce the number of photons, your Einstein will turn back to be Marilyn in some way. However, what is relevant is the new way data are proposed to you. You need instruments. This is the reason why we try to build them. And this is Marilyn, and this is Einstein. Now, believe me. The point is that the medical doctor today, or biologist, you have to take a decision about what's going on using this image. You can be wrong. And then you can say, okay, guys, this is what I had. Well, if you switch here, you can start counting molecules. You can be wrong. Maybe you design not in the best way you expect. You have like excuses today, and you can get more information. So I don't know if you remember about the fact that the term microscope has been coined in Italy for the Galilei microscope. Doesn't matter if it was the first or not, but the term was coined in Italy. And today I think that we can use, instead of microscopium nominare libuit, nanoscopium nominare libuit, and we are in the nanoscopy domain. Because today we are able to move from 100 down. Instruments. How can we do with a single molecule? This is your system. There are the molecules. They are not photoactivated. You photoactivate some molecules. The distance is larger than the diffraction limit. You photoactivate them. Then you excite so you can see them. You bring them to photobleach so they disappear. And every time you evaluate the location of the molecule with the rule said before, d divided by square root of n, more or less. And then you bring your final image. Even if your final image is similar to the conventional image, the information that you have now are the position of all these molecules with the precision of 10, 15 nanometers, not 200. And so you have to repeat this game. This is your original image. This is your particle detection, if you ask them to shine light not all together. And this is your reconstruction and final image with a very high precision of localization. This was the seminal paper by Betzig. And please have a look here. Submitted March 2006, accepted August, published August. And in this paper, he was bringing to your attention that fact that your localization intensity is scaled with the square root of n. When you have 200 photons collected by a single molecule, you get this in terms of position. When they are 800, you get this and this. And the experiment was this one. Having the sample, photoactivating, exciting, collecting photons, bringing them to photobleach, and putting the information in your picture here. And this is the precision. Here, I don't mind about all these terms. This means which is the diameter of this circle. N is the number of photons. Unfortunately, even if n is large, if you have B large, large is background signal. You are lost. This is one of the points. Again, so this is the construction of that image that you can see here. Sam S submitted in June and published on Biophysical Journal in December, having the acceptation in August, but later than Betzig. More or less the same paper. Shouwei Zhuang submitted in July and was published online in August, before Betzig, but was submitted later. Shouwei Zhuang did a different game. Not bright, dark, but red and green. Doesn't matter. What is relevant is that you are able to control two states. And so this is the result, and this is what you have to pay. Collecting 40,000 frames and localizing all these points. Now is upon you. If you have a question and you need this, so if you, from this information, you cannot say anything, but you can say something from this information, well, play this game. When you play this game, you have to take care. Because you have, these are your molecules. This is the localization done in a very good way. And this is what you can get. But now, if you don't collect too many photons, and so your circle of indecision is large, what you get is something like this that is not exactly this one. Or in case you are from Genova and you decide not to photoactivate too many, because there is a cost associated, and you photoactivate less, you have this signature that is not this one. But then when you are in the real case, molecules are moving, and you have to take care of this. This allowed to develop a very nice trend in particle tracking. So if you are great in particle tracking, you can also use this information with moving molecules, getting super resolution. And this is the case when you collect a high number of frames, high number of photons and of frames, and this is the cost you have, and this is what you can get. Again, you build an image in this way, like a pointillist painter. Sorry. Now, let me back to this. There is one point now. I have a question for you. When do you stop collecting photons? When?
When the object stops emitting the specimen.
Okay, do we have a more quantitative criterion? You could say, when I recognize something, it's not good because this seems that you cannot discover anything new.
Fixed or moving set?
Fixed sample. We don't mind. The answer is in the next slide. Yes, if you know what you... Now, but your answer is correct. Simply change a little bit your answer. When I get the resolution I needed to answer my question. Is fine? When? When is satisfied a criterion called the Nyquist or Shannon criterion that tells you that if you want to have 100 nanometer, you have to sample your sample at 50 nanometers, so 1/2. So when you have a large amount of data of localized molecules that are at distances 1/2 of the resolution you want, you can stop. Could be a good criterion. You will find this in the notes that I distributed, even if late, I'm sorry. And so you can work on this. It is a stop criterion. Or you can stop when you recognize the structure. But this is the reason why people is working on filaments. They immediately recognize the structure, and then they can get a lot of interesting information, also in terms of resolution. And there is a bad way of measuring resolution. I don't want to discuss too much about this, but you can decide that your resolution is the full width al maximum of the filament you are imaging, or the minimum distance between two filaments, or whatever you want. Is open this, like Toraldo di Francia was predicting. Again, this, or this that we collected, Francesca Cella collected with the N storm. And you can get information along the z-axis. Now, I don't have too much time now, so I would like to switch to another method, but I want to show you only, okay, let me, in case I will go fast. I'm interested, I'm getting old. I look back to literature, and I find so many papers related to fluorescence and related to cells that lie on a cover slip, on glass, that I'm thinking, maybe there is something wrong since the beginning. We are studying interaction between cells and glass. That's nice with a glass of beer, but for the single cells, I'm not sure. And so we are moving to aggregates of cells. When you have aggregate of cells like this one, like this one, the problem you have is that in case you try to perform single molecule localization, you have a lot of background noise. B is very high. In order to cut this noise, I will jump to the last slide now on this topic, we decided to use not the conventional way for collecting information, so the lazy way. I shine light, I collect light. The sports way. Shine light, I move fast, I collect light 90 degrees apart. Okay. In this way, I'm cutting the object with a plane of illumination. I have background here, but I don't have background here. And so my B is less. And so let me through this. If I play this game on the cellular aggregate, I can navigate in my sample with a plane of illumination, getting single molecules, and so getting the possibility, we can discuss about 3D if you are interested, and deciding in which region I'm interested in the super resolution, and then I can get a super resolved image in a region of a small object. This is our main interest today. If I use two photon photoactivation, my plane of photoactivation does not suffer from scattering, or suffers less. And this is what we did again. Okay, 2 minutes for this. 3D, time, I want to be fast. I have a problem in being fast. I have to move a glass object, a lens. I have to push. So there is some inertia. We decided to switch to a device that is this one, this liquid, that is a liquid lens. There is a liquid that is in this tube, like San Gennaro blood. You plug this on the back focal plane of your objective. Any microscope you have, doesn't matter. And then you send some music, not audible music. I mean, from 140 kilohertz up to 9 megahertz. What does it happen when you send music to a liquid? It compresses and decompresses. But compression and decompression produces a change of a molecular property that is a refractive index. And so there is a change of focus in your sample, like the one you have when you're moving the lens. But you can be very fast. You can be so fast, and you don't mind having any synchronization with your system, that even if your object is moving, you can find it always in focus at a certain time. Because you're so fast, you're driving in a so fast way your focus, that there is a region where the object is collected in focus, even if it's moving fast. So we tried first with this, let me say, easy sample, because there is a lot of fluorescence. This is what you can get with a single plane. In the time we get a single plane, now we are able to get 64 planes. So you simply perform your scanning, and while you're scanning, you are collecting 64 planes. Then decided to put this lens in the selective plane of illumination to try to have this faster, because you have to move this plane of illumination in your sample. And we decided to use a very fast camera. In case you are slow, you can record traces from particles, not fluorescent. These are scattering particles, because for fluorescence signal is too low. This is a proof of principle. But what you can do, you can collect at this rate what's going on. Not this rate, better, this rate. So you can really track in a very, very fast way what's going on in your system. We aim now to bring this to super resolution, where you have a very low signal. This is the main problem. But you can play this game. So this was a first attempt, and also this one, to have direct 4-dimensional imaging of the sample using this fast movement of the lens. Now I'm on the last part, and this is about STED, about Stefanel. STED, this game, is comparatively simple. You excite, you get fluorescence, and you detect, usually you detect this peak of emission. You could also play here, but why not working here? Now, when your molecules are here, ready to fluorescence, you shine a second beam that brings them down to the ground state without waiting for their natural decay using fluorescence. You can cancel some fluorescence. And one is when you don't apply any second beam, you have your all fluorescence. Then you start shining a second beam, and so you are killing some fluorescence because you are convincing the molecule to deactivate in another pathway. When you increase this second beam, your signal decays in this way. You can call this, when you are 1/2, so-called intensity of saturation towards 0. And this is in a number, is a property that is specific for your fluorescent molecule. So you have a molecule, you excite this molecule, you bring to the excited state, and then if you apply a certain intensity to the very same molecule at another wavelength, more or less here, and there is a reason. You apply here because this is a very intense beam. And if you apply, you could apply also this intense beam here. The point is that you are too close with the absorption of your molecule. And since this is very intense, you have the other effect. So you are killing some fluorescent that you excited, and you are priming other fluorescence. So you don't want this. And so this is the game. Pictorially, what happens is that when you increase the intensity, you have this hole very, very small. So you are again a pointillist painter, different school, because now we have a hyper-technological brush that is very, very thin. Before you were collecting in a very precise way information from one point, watching and then trying to define the exact position, whatever. Now you have a brush that is very, very, very thin. That is, instead of this one, is this one. Okay. Again, pointillist painters, Pellizzara Volpedo. What I like in this method is that I don't need 2,000 or 40,000 frames. Even if there are, for some aspects, I like the other method. It depends on the question you have. In this case, this is your conventional image. Then you switch on the second beam, and you immediately get this. You don't need any computation. You don't need anything. When you go into the cell, it's more or less the same. Wherever you shine your second beam, you get an improvement in resolution. And also in this case, I will be on this later if I have time. And you can play this game real time. I mean, real time as real time is confocal microscopy. Again, when we are here, you can get this kind of information. Just a few examples, also in the third dimension. And now let me be on this number. Responsible of this is a ratio. Is the ratio between your strength, the intensity of your donut beam, and a photophysical property of your molecule. So now, if you have a good friend in biochemistry, able to produce for you a molecule having a very low IS, you can get a very high ratio with a very poor beam intensity, with this laser pointer. So you need this. These are the developments in fluorescent molecules today. And so this is the way you scale. Now, since it's not very clean, I don't know why, I think that we are authorized to have some act of vandalism on this with some spray. And so with some spray, you can add this term. This term tells you that this is the Abbe formula, resolution formula, call it as you want, because Abbe never wrote this formula. When you see, say, I don't know if you can read German. If you can, go to your library, take the paper that everybody is citing with the Abbe law, and try to find this formula and experiment dealing with the minimal distance between 2 point-like sources. It's not in that paper. However, the formula can be derived from that, and it works. Now, you can add this term. I is your strength. IS is the property of the fluorescent molecule. Toraldo di Francia is the a priori knowledge you need. And when I is 0, so you don't apply the second beam, you have the conventional formula. That's it. and is unlimited. There is, if you can bring this to a very high number, to infinity, this will become 0. If you can collect in the other method an infinite number of photons, for example, with a nitrogen vacancy in diamond, resolution can be 0. And in this case, it depends on the intensity of the second beam. Now you have to take care, because this second beam is true that is in the infrared. It's not true that in the infrared your sample is not absorbing at all. It has a very poor cross-section. But since you are using a large amount of light, something happens. And when something happens with a fluorescent molecule, this means that the molecule emits. And if the molecule emits, then like you, that you would like to stop listening, the molecule will stop fluorescing. And if your molecule, while you are playing this game, photobleaches, you have lost your idea of resolution, because you have lost you don't know if you have lost or if you are the molecules because they are there or not. So you don't want this. So we have to find a way for reducing this. But let you know that when this ratio is 90, you can get 16 nanometer. When this ratio is 300, you can get 8. And in principle, you could localize with an atomic precision. Are we interested? Well, we are working room temperature with living systems. We know from physics textbooks that there is a constant called Boltzmann constant that when multiplied with temperature makes something to our system. And so even if we push to the atomic possibility, we have a system that is moving, that is changing so fast in space that maybe is not so relevant. 30 nanometer is a good compromise. But you can push in a limited way. This is the best resolution achieved, 2.4 nanometer with an optical microscope. Betzig, against this method, raised this problem with the intensity, what I was telling you before, and L decided to reply in a very nice way, even if I don't like this second method. Can I take just 3 minutes more? With your second beam, the game you're trying to do is to have a so-called stimulated emission that has a very poor cross-section, so you need a very high intensity. But if you have photoswitchable molecules, you can use the donut beam for promoting the switching. And so they are emitting in green, but then the ones switched are dark or red, and so you don't read them. I'm a little bit against this method because it's too much sensitive to the environment. So I'm not so sure that I'm able to define the right intensity of the donut beam for having every point the very same resolution, but it doesn't matter now. And the time scale is different. For simulated emission is the picosecond. For resolved or this game, for switching games, is in the microsecond. So it's a different game. However, there was this interesting paper years ago by Stefan and others. And Ilaria Testa, that was in our lab, then moved to Stefan, had this nice experiment with photoswitchable molecule, reducing the light levels for getting super resolution in living brain slices. And this is a paper that you can easily find is open access. So this is the game, but why we are interested in reducing the donut beam intensity? Technically now, not from the side of biological sample. I'm interested, but not so much. But I'm from Genova. I would like to be fast. In order to be fast, I could parallelize. So I could have more donut at the very same time. But if I need a very high intensity and I have to split this in several donuts, I really need a laser that is not available. So if you are able to play to reduce the intensity needed for having the very same resolution, you can play a game parallelizing or patterning your STED in the sample, being faster. The last few slides are about a method for reducing the donut beam intensity, getting the very same resolution. So you need 600 milliwatt for getting, I don't know, 40 nanometers. And now you need, with this method, 50 milliwatt. So 1 order of magnitude less. Because now I'm using another information coming from fluorescence, lifetime. I don't know how many of you performed fret experiments. But when you perform a fret experiment, you can find that you are in the fret condition measuring lifetime, that your expectation is that it's faster. For a very simple reason that your molecule has normally the possibility to deactivate in a non-radiative way, deactivate by fluorescing, deactivate by fretting. And so lifetime is shorter. Now can deactivate by stimulated emission. This means that even if the intensity in the donut is not very high, and let me graphically think that some photons are able to pass through the donut, which is the difference between these photons and the photons passing through the center where I do nothing, because these are the photons I want to see. The very first one have their own lifetime. The other ones have a faster lifetime. So I shine excitation, I count time, photons arriving too early are excluded. And so what you can get is a STED with 1 order of magnitude less intensity of the donut beam, and the very same result you can get with a conventional STED, with a so-called gated STED. You can play this game to photon with fast-gated detectors, but since the driving force is lifetime, and Luca Lanzano is from Sicily, but he works in Genova. And so my question was, why, Luca, we are losing so many photons at the end? Because we are not collecting photons that are available. Can they tell us something? Well, let's classify arrival of photons with their lifetime. If you do this, so encoding and decoding spatiotemporal information, you have this situation. This is the donut. Photons coming from here have a certain lifetime. From here, a different lifetime. From this region, another lifetime. If you keep them, and if you classify them, they come. So, in some way, you can have 3 interesting images at your disposal. One is the super resolved. The other one is the confocal-like. But the third one is the background. Background is some uncorrelated signal, photons coming that are not related with your process of shining light at a certain time. But it's relevant because when you perform super resolution, you have a low budget of photon coming because you are super resolving, so you are collecting few photons from few regions. So if you are able to remove background, you can get an advantage, like in this case, the method is called the split. And this is the advantage you get with respect to the gated and with the confocal. If you are interested, here you can find 2 nice reviews on the STED. And since here probably you also have atomic force microscopes, think to couple this method with an atomic force microscope. Why? Because the atomic force microscope is high resolution. Unfortunately, it's a surface method, but can tell you something about 3D at the surface. But these are specific. Fluorescence is highly specific. Okay, but when you try to couple them in this resolution shape, you don't get too many information, but when you are very precise with your fluorescence, you can also be more precise with your FM topography, and you can correlate your atomic force image with a fluorescence image in a better way. This is the correlation you have with STED FM, and this with a confocal. You can use this information for moving your samples or for doing whatever you want with your sample. But what I'm interested in is that when I'm trying to perform some force spectroscopy here to say something about mechanics, nanomechanics, I want to know how many filaments are there. I want to have a better resolution there. Or this is the work done by Christina Flores on DNA, or this one on ribosomes. So we are starting coupling this method to get advantage of the specificity of fluorescence and of the high resolution at the surface of the atomic force microscope. And I would like to thank you for your kind attention, and sorry for being so late.